So I'll just reintroduce myself again. So I'm Tilo Kunath. I'm a researcher at the Center for Regenerative Medicine in Edinburgh, and a lot of you have been there. I know I can see a lot of familiar and friendly faces. I'm going to give a little summary of 3 talks from the Congress. So I didn't go to the Congress myself, but Nicola Drummond in my lab went to the Congress and presented a poster and gave us a 2.5-hour debriefing a few weeks ago. A very detailed description of a lot of the Congress. But I'm going to really pick on 3 talks about clinical trials and things that are very close to clinic, because I don't want to hurt your brain by talking about all the intense science that Nicola was reporting back to us. So I'll see if I can use this. Yeah. So the first talk I'm going to speak about is actually related to the last video. So those 2 videos were amazing. I really love these videos. But this young woman was talking about deep brain stimulation. So there was one session on deep brain stimulation by a professor in Toronto, Andreas Lozano. So he's probably the world leader in deep brain stimulation research. And he's been using it for a long time in Toronto, and he sets the stage for a lot of the clinical application of this technique. So I don't know a lot about this technique. It's a neurosurgical technique. It's usually used to control motor symptoms in the later stages of Parkinson's. It's an invasive technique, of course, because it involves a neurosurgery, and you have electrodes implanted in your brain, and you have a battery pack around your collarbone that you need changed. But a lot of people have had this, right? 150,000 people have had deep brain stimulation, and it's about 10,000 a year. Does anyone know how many people in Scotland get DBS a year? It's close to 0. Yeah, I don't think it's offered in Scotland.
It happens in London.
Yes, exactly. So you have to go. Yeah. So Gavin Hastings' wife, I think, has had DBS, and she lives in Scotland. People in Scotland can get DBS, but it's not going to happen in Scotland. You'll need to travel down south.
There's supposed to be a surgery centre opening in Glasgow in the near future for DBS.
Yeah, that's good to know. And there's also going to be one in Edinburgh. There's also some news I was going to share as well. So the Glasgow one I didn't know, but there's serious efforts to at least get some capacity for DBS in Scotland closer to home, which isn't happening now. And I don't know if you've seen videos online about what this procedure can do to improve motor symptoms, but I've watched some YouTube videos. It just looks phenomenal, and it's instant. It is instant, right? So the stem cell therapy I'm going to speak about shortly takes 2 to 3 years to kick in and actually work. While DBS, when it works well, you can come out the next day or the same day and have your motor symptoms corrected with this technique. So, the best outcomes when it works well, it looks amazing, right? It looks fantastic. You can control a lot of the motor symptoms, the fluctuations, tremor, gait, posture. So a lot of the quality of life things can be fixed with DBS, but a number of them can't. So you heard also that the young lady had all these gastrointestinal problems. Those won't be addressed. Sleeping problems, progression towards dementia, depression, all these non-motor symptoms are not addressed by DBS. But a lot of the motor, or almost all the motor symptoms, can be addressed by DBS for some time. So this is just, I'll show you something that Andres is doing that's more advanced, but this is a therapy that exists in the world today. Some people are getting it and doing really well, but just locally or nationally in Scotland, there's very, very little access to it. Right? So I don't know if this is a point that Patrick and David want to bring up for advocacy in the next few years. So he wasn't speaking about DBS because it's an established technique. He was speaking about it, but he didn't harp on about it. But what he is known for is pushing the limits of research with this technique. So he's applying DBS for Alzheimer's as well. But he's now moved and published something in 2016. Something called MRI-guided focus ultrasound. So before Nicola presented this to us, I've never heard of this. So you know about ultrasound, right? So when usually it's used during pregnancy to look at the unborn child, but it's used for a number of other indications, and you could use really high-intensity ultrasound to ablate or to kill some neurons. And they've used MRI to guide these trans-skull penetrating ultrasound. So there's no surgery, first of all. That's a major difference between DBS. And I haven't read the paper yet, but in 40 individuals, mostly with essential tremor, they had some amazing results. And I think these videos will work. I'm actually going to ask if you could play them. So this is Andreas there, but this is a gentleman with very severe tremor. And actually, if there's sound, can you put up the sound?
You're not going to be able to do it. Okay. Let's. Okay.
So this is him the same day of the procedure.
After several hours, Tony is brought out for his post-procedure assessment, where the effect is crystal clear.
When's the last time you could do that?
More than 10 years ago.
So pretty dramatic stuff. So, N of 1. I don't know how this relates to every person that gets this MRI guided ultrasound, but you know, if you can have effects like that, even with DBS alone, an established technique, same day improvement of motor symptoms, I think it's something worthwhile getting access to. And it is, I think it is a bit of a shame that, well, it is a big shame that it's not more prevalent in Scotland. Okay, let's see. Okay, so the next, yes, yeah, we can talk about this because I'm going to completely change subject now.
When will it become more prevalent in Scotland?
Say it again?
When will it become more prevalent?
DBS,
in Scotland? No, not DBS, the guided ultrasound.
This is brand new, so he's probably the only one in the world doing the MRI guided ultrasound. How quickly it gets taken up by other clinics across the world, I don't know. This is really far outside my field. I don't normally read about DBS, and again, this is one patient as well, so always be wary about one patient doing well. Right, and they somehow knew he was going to do well because they were filming him. You saw all these cameras before he went into the surgery, right? All the press were there before he went into the, well, I shouldn't say surgery, for the ultrasound, the MRI guided ultrasound, and the press were there when he was coming out. But they must have had some early success to know that the region of the brain that they're targeting would improve his motor symptoms. So I think these individuals were people with essential tremor, so people with more sporadic Parkinson's. I don't know yet if he's treating these individuals. Yes?
My question was just, do we know yet how long the effect lasts? Because it strikes me you can't go through ultrasound after ultrasound if it only lasts for a few weeks or something.
No, yeah, I think it's a long-lasting effect. The way that it's a believe it or not, it's a technique to kill off some neurons. And I think they're killing off neurons in the subthalamic nucleus. So they're actually using ultrasound at such a high frequency. When you use it for looking at pregnancies, the ultrasound is very, very low frequency, so you can get images of the baby. But in this method, he's using a very high frequency ultrasound. He's actually destroying a part of the brain that he knows from his deep brain stimulation studies that will improve the motor symptoms. So it's permanent. Wow. It's not going to be, you need an ultrasound again 3 weeks later, etc. It's not a temporary thing. Yeah. But as you know, I didn't go through all the downsides of DBS. There are some, and the DBS only works for some amount of time. And again, I don't know this surgical technique very well, but there are pros and cons to DBS. And one of the biggest cons, though, is that it is an invasive neurosurgery, right? So if you can remove that part of the equation by having an MRI-guided ultrasound, that removes a lot of the risk of surgery, infection, and recovery, etc. And no batteries to be changed once a year. Yes.
How long does the MRI treatment take to do?
Sounds like it takes an hour, maybe less. Yeah, I don't think it takes a long time. They'd probably need a stereotactic frame. You have to go into an MRI. They need to know the precise region of the brain that they want to target. And then you would have probably some ultrasounds coming from different directions, but the focal point where you want to kill neurons is going to have the highest level of damage, and then, like that video was hours after their procedure. He doesn't have any stitching up of the skull, and so it sounds magical, and if it could be brought more widespread quickly, that would be fantastic, and if it could be proven in more typical Parkinson's, that would be fantastic as well. So I've met him, and he's an outstanding clinician. So I've met him in Toronto, and he's a really solid guy. And I think even though we're showing one patient there, and the paper will have variable results, but I would really trust what he's showing there. Yeah. Does it help what? Mobility. The MRI guided ultrasound. I don't know yet. Yeah, I don't know. Yeah. Yeah, DBS does. So presumably, if he's trying to target the same neural pathways that DBS targets, then yes. And I think what it won't help, again, DBS doesn't help, are the non-motor symptoms. I think it won't help constipation, sleep conditions, pain. Yeah. But I think it's very exciting, and I'm going to get Gordon Duncan to look into it more, because he understands all the clinical side of Parkinson's. Andreas Lozano. I can go back, yeah. He's at the Western General Hospital in Toronto. Yeah. Now, one of his trainees is a good friend of mine, and we're trying to get them interested. We're trying to set up some collaborations for stem cell transplantations with Toronto and Saskatoon. So, yeah, and I'm very fortunate to have a neurosurgeon in my lab now, so he understands all of this stuff. But yeah, I think it's exciting. So that's talk number 1. Yeah, okay. These slides are all available to anyone that wants them. I'm going to leave them with Heather, or I can leave them with you, David, and just distribute to anyone. There's nothing confidential in these slides. Stick it on the website, yeah. So the next talk is actually in a parallel session from a very well-known scientist named Eliezer Maslia. He's quite a very senior scientist. He's been in the Parkinson's field for a long time, and he seems to have his finger in lots of near clinic treatments. Like he's really working at the cutting edge between basic science and getting treatments into patients. Right? So he gave more or less an overview talk of a lot of treatments that are in phase 1 clinical trials, phase 2 clinical trials, and preclinical. So I'm only going to talk about 3 trials that are all based around this protein called alpha-synuclein that are either in phase 1 or beyond, meaning that some patients in the US are getting these 3 treatments that I'm going to talk about. And they're all around this protein alpha-synuclein that I could talk for ages on, but I'm not going to. An alpha-synuclein vaccine, something called passive immunization, and then he talked about a number of novel small molecules, which is just a fancy name for drugs, like chemicals, but I'm just going to speak about one of them. Okay, so this is the first trial that's been going on in the United States by a company from Austria, actually, Afarius, and it's co-funded by the Michael J. Fox Foundation. So even I had to look at what's the difference between active immunization and passive immunization. What's the difference? So active immunization is something that happens usually when you get a vaccine or flu vaccine that you may have got recently, where you have dead virus or immobilized or incapacitated virus injected or inhaled, and your body makes antibodies against it. Your body reacts to it. So that's active immunization. This is the normal immunization that you think about. So this company has made a small bit of alpha-synuclein, a small bit of peptide, and they have attached to this something that they don't tell you what they have attached to, something proprietary. And then people get injected with this, like a vaccine. And then this attachment somehow makes the T cells not react, but the B cells, which make antibodies, do react, and they start making antibodies against alpha-synuclein, like it was a virus. Right? So they inject this thing, they're calling it PD01 afitope. So I'm trying to dig into the literature. What is this thing, right? I go on their website, and it's a bit hidden. It's a bit proprietary, but what I see that they've made a small piece of the protein. The protein is 140 amino acids, so they've taken a small piece of it, and they've done something to it, and they've made a concoction that they're injecting into people. Right? And they've done a lot of preclinical testing in mice. It reduces the amount of alpha-syn in the brain. It reduces neurodegeneration, dopaminergic loss, again, all in mice. And it promotes the clearance of alpha-synuclein. So alpha-synuclein is this protein that makes Lewy bodies and is pretty much considered to be the driver of Parkinson's disease. So these drugs that people are coming out with now are attacking what I consider the heart that's driving Parkinson's disease. Well, levodopa and even DBS treat the symptoms, right? But this is trying to stop the progression of Parkinson's disease in its tracks. If you can stop alpha-synuclein, right? So they did, so far, it was reported a phase 1 clinical trial, only 12 patients, but essentially that phase 1 trial means it's a safety trial. Is this a dangerous drug? Is it okay to give to people? There were also, I think, 8 volunteers, so 20 people were given this, and nothing really bad happened, which is good. 50% for some reason, 50% did get, so there should be antibodies, did raise antibodies to alpha-synuclein in their blood and in the cerebrospinal fluid. So I'm not sure why half of them did and half of them didn't, but the effect to get antibodies raised against it did happen in half of them. And now phase 2 means it's an efficacy trial. This will actually slow down Parkinson's. So they're in that now. Phase 2A, PD. So this is one trial that's happening now, and they also have a second trial looking at multiple system atrophy, which is a rare condition, but it's absolutely related to alpha-synuclein. Dementia with Lewy bodies could also be another trial that this therapy could be used for. So these are in phase 2 clinical trials. You can look them up on clinicaltrials.gov and then keep an eye on press releases from these companies. But these are in people at the moment and in clinical trials, currently just in the United States.
Commonly stated that the involvement of alpha-synuclein is through misfolding.
Yes.
So is this vaccine something that only affects the misfolded form of alpha-synuclein or not?
Yeah. So this one, no. This one affects all alpha-synuclein. And what happens in Parkinson's, you're right, there's misfolding of the shape of it is not correct, but it also accumulates. When it's misfolded, it doesn't clear from the cells as quickly as it should do. Everyone has alpha-synuclein. We all have a lot of it, actually. Alpha-synuclein is an abundant protein in human brain, just in general. But in Parkinson's, it misfolds, and it does accumulate more than it should. So clearings, this is why they like to see that there's a promotion of clearance of alpha-synuclein. So just reducing the levels of alpha-synuclein is beneficial. So it's addressing all of it. And you might ask, is alpha-synuclein important, though, for normal function? And as far as everyone can tell, the answer is no. Then you might ask, why do we have alpha-synuclein? What's it for? So we don't know what alpha-synuclein is for normally. Mice have it, all mammals have it, and you can remove it from mice, and the mice are fine. Right? So we don't know if it's doing anything important in normal function. It's certainly doing a very bad, has a very bad side. Right? Yeah, so that's active immunization, and I think they do hide it a little bit because it's a bit scary sounding. You're injecting a protein that's actually known to cause Parkinson's, right? If you take the whole protein, 140 amino acids, you inject it into a mouse, that mouse gets Parkinson's, right? So to inject something that looks like alpha-synuclein sounds a bit scary. So I think this is why you almost, it took me a long time to even understand that it was a small peptide of alpha-synuclein itself, because they just use this term, PD01A, which means nothing, right? Okay, so passive immunization is very different. This is a much newer thing, which is also, to me, sounds much safer. So passive immunization is where they actually make the antibodies outside of the person. They make it in the lab, and then you get infused antibodies. It's the antibody that's doing the clearing of alpha-synuclein in the active one, but the antibodies are made by yourself because you were injected with this, and then your B cells make antibodies. So passive immunization is that the antibody is made by the company or the lab, and then you have an infusion of antibody. This is becoming really popular now for arthritises, and it's a very novel way of using a therapy. But this is so it's not really a vaccine, but this company Prothena was the one that really pushed the technology forward, and they were bought up by Roche, and they had a mouse antibody called 9E4 that was working really well in animal studies, but it's very problematic to inject a mouse antibody into humans, so they have this technique that they call humanizing. So they humanized this mouse antibody, and this drug now is called PRX002, and it recognizes a very small portion of alpha-synuclein. So these little Y-shaped structures here will see alpha-synuclein and will bind it in the region between position 118 and 126. So I mentioned alpha-synuclein is 140 amino acids. This antibody will see it almost near the end. And then what it's hoped to do is to clear it, is to get rid of it, right? We want to reduce alpha-synuclein to stop the progression of Parkinson's. And they also have been through a phase one clinical trial. 30 patients, this antibody was safe and well tolerated. One of the main measures that you want to test is alpha-synuclein reduced. It was reduced in the blood after a single administration, which was very good. So they're now in phase 1B, which is still a safety trial more or less, but they're increasing the dose of this antibody in Parkinson's patients. And then if that's all fine and safe, they'll go to a phase 2 efficacy trial. So this is another exciting, really disease-modifying like therapy that addresses the engine that's driving Parkinson's that is nearing really big clinical trial. So we'll need a phase 3 double-blind clinical trial before it can be distributed out for the world. But I think these two are both very promising therapies to slow or halt the progression of Parkinson's. Okay, and yeah, so this is just one of this, because a lot of people can make antibodies like this. A lot of companies now are very good at making these antibodies, and there's lots of other groups that are reporting that these anti-alpha-synuclein antibodies can protect against dopaminergic loss. So the previous drug I showed you is very specific to that one company in Vienna, right? And that's okay, but you always like to see, as a scientist, you want to see other labs doing the same thing and getting the same results, right? So this type of approach of making an antibody is working in other labs and other small companies as well. So I think it's a generally accepted phenomenon that this passive immunization looks really, really promising. Okay, the third drug that has been in clinical trials that Elijah spoke about is a small molecule now. So this is actually a drug. This is a drug that's orally that you can take. So the antibody treatment needs to be IV. This is a drug that I don't have a structure of. This is hidden. This drug, MPT 211, can't find it on the internet. But there's a similar drug, MPT 118A, that looks like this. And I think this drug will be quite similar to that, but not identical. But there's been a very nice paper published on this drug, showing again that it reduces alpha-synuclein, it reduces its toxicity, and it rescues animal models. So this drug here has been in phase 1 trials, 8 patients, and they just released the data. It was all very safe and well tolerated, and now they're approaching phase 2. And this drug came out of a small company called Neuropor that's just had a $60 million buyout by UCB. So UCB funds 2 projects in my lab, actually. They're very, very serious about this drug here, and it looks, all the animal data looks really promising. And the safety data in animals looks great, and the safety data from this trial looks really good. It looks really a safe-looking drug. And it's a big issue for drugs like this because it's a completely new chemical entity. This type of molecule has never been in man, so that really scares regulatory agencies. Is this going to be a safe drug? Is it going to have side effects? So far, a little bit of data says it doesn't.
One of the things that we brought back from the conference was the realization that it's not just dopamine, that there are 4 neurotransmitters involved. Mm-hmm. Now, do these drugs, does the alpha-synuclein attack all 4 of the neurotransmitters, and do these drugs work for all of them?
Yeah, so that's a very good question. So these drugs are attacking something well before neurodegeneration is happening for any neurons, right? So cholinergic neurons, as you say, dopaminergic neurons, so a lot of these neurons are dying, but they're all dying for the same reason. They're all being killed by alpha-synuclein. So this drug is acting these drugs are acting at a completely different level to where dopamine and cholinergic agonists are acting. So these are acting to replace the neurons the activity of neurons that are lost, right? These drugs are acting to prevent the neurons from dying at all, right? Or prevent the neurodegeneration from continuing, right? So it's the point that it's acting is totally different, right? So it's acting at the mechanism of neuronal death, while all the other therapies now are just trying to not cover up, but make do, try to address the symptoms, if you know what I mean. Yeah. So these type of drugs don't exist yet. There's not a disease-modifying therapy that slows down Parkinson's in a very reliable way. These efforts from this guy, Elijah, has been really, we want to stop the progression from going any further. Right? So they'll be using combination, of course, right? So if, say the Parkinson's can be slowed dramatically, but you'll still need the L-DOPA. You'll still need the medications that you'll be taking. But then the hope is that the condition won't progress so that you need more medications and more complicated regimes, et cetera, right? But yeah, I'm going to speak about the last project the last talk in a minute. But this is just to give you a flavor. And he did give a summary of more drugs that are closer to phase 1. So these are the 3 that I just mentioned, backed by different companies. These 2 very big companies, Roche and UCB. This is a small company, but backed by this charity, Michael J. Fox Foundation. So this is approaching phase 2. There are other companies that are attacking alpha-synuclein, neurophage with a very unusual molecule, and then a second passive immunization. So passive immunization is, again, when you make the antibody in the lab and then you administer the antibody. So here's a second passive immunization program. And Biogen is a fairly big company, so I'd imagine this will be in phase 1 soon. Yeah, it's a very good question. So the passive immunization ones, I think, will not be very slow. So this PXR002, they're called biologics, meaning that they're an antibody, and antibodies are being used now quite a bit. You need an industry person to get a timeline. But I think if phase 3 went well, it would be a matter of months/years, I think, for a biologic. For a new chemical entity like MPT-211, it's a brand new drug. I think they'd be a lot more cautious before giving it to a lot of people, because it's a drug that's never been in man. Maybe it has very severe side effects. So I think for a small molecule, they're going to be a lot more cautious. Active immunization, this one, I really have no idea. This is such a new concept for neurodegeneration that, I don't know how long that would take, but yeah. It's not, these are, of course, much closer than stuff that's in the preclinical stages, which I haven't spoken about today, which will be quite far. Yeah. Yes?
I would like your comment on this, because you mentioned dementia, I think, and the relationship of this protein to that. I have had Parkinson's for 14 years and a bit more, I think. My husband developed dementia after 31 years of marriage and is now in a care home. I think it had been coming on for him for about 10 years. Mm-hmm. Can you comment on the relationships between the illnesses and what might be influencing each other?
You're asking why you got Parkinson's, he got dementia?
Well, yeah. Is there any relationship between them?
There is a molecular relationship between Parkinson's and Lewy body dementia. Not sure if your husband had Alzheimer's or he may not know actually. His clinicians may not know either. But if we're talking about alpha-synuclein, The major alpha-synuclein condition is Parkinson's, but multiple system atrophy and Lewy body dementia are also caused by alpha-synuclein, which is why actually all of these drugs would be useful for MSA and Lewy body dementia, because the alpha-synuclein is at the heart of that. And there are other rarer conditions that alpha-synuclein causes as well, and all of these would be useful for that as well, right? So, if they can diagnose someone that has Lewy body dementia, and it's sometimes, I think it's not that easy. If Gordon Duncan was here, he could help me, but I think Parkinson's dementia, this could also help that. So preventing the spread from just a motor condition to a motor condition with dementia, this could help there. So DBS, for example, is not going to help the progression towards dementia. It's not going to stop that. But these drugs here could. Because the idea that the motor symptoms turn into or move to a situation where the frontal cortex is affected, getting dementia is very scary, right? So if you can stop your Parkinson's to being just a motor condition instead of a motor and dementia condition with these drugs, that would be fantastic, right? So that doesn't quite answer your question, but these drugs will affect, so I should make the point, stopping alpha-synuclein will affect the motor and the non-motor symptoms. That's the main message of big difference between deep brain stimulations and what's up here, right? And the next therapy I'm going to speak about, stem cell therapies, as I'm going to describe them, just address the motor conditions. They won't address dementia and the non-motor conditions, stem cell therapies. Okay? You're going to mention Lisa Davenport? Absolutely. Yeah. We need, I think a lot of these clinical trials, I think all of them, these really exciting ones, are happening in the United States. How do we get a much quicker connection or tie-in with what's happening in the United States to get drugs over here, right? And there are ways, I think Biogen is interacting with Siddharthan, doing a bespoke clinical trial in the Ann Rowling Clinic right now. So there are ways to entice these big American trials to put an arm in Scotland or in Edinburgh. Yes?
They're based in America, that the new president's going to make any effect on what's happening?
Yeah, I don't know. Yeah, time will tell. Yeah, and we'll Belgium, that's why UCB is based in Belgium. They have a really big setup in Slough, and they're also working on a passive immunization therapy. It's not up here because it's further from clinical trials, but because they're in Belgium, will Brexit have an effect on their Slough setup? Will they pull out of Slough and take all their stuff to Belgium, and then we have less quick access? Sorry? In the Netherlands? Oh, I mean interactions with Bastian Bloom and stuff like that. Yeah. It's I'm going to ignore all these geopolitical problems that may. I'm going to go through the last session. I'm not sure how am I doing for time. Yeah. Carry on. I'll just carry on. So the last session is something I do know a lot about. I think this is not working. So half of my lab works on a cell replacement therapy, and my background is as a stem cell scientist. So Roger Barker gave a talk on the history of cell replacement therapy and where it's at today. So I don't know how many of you are familiar with cell replacement therapy, but it's this idea of replacing the dopaminergic neurons that have been lost in this striatum with a neurosurgery, right? So that levodopa works really well because the dopaminergic neurons are dying, so that levodopa is very good at compensating for that for a while. But the idea of cell replacement is can you regrow dopaminergic neurons in the brain? And that's been tried since the late 80s. And the ideas have been around since the 1970s that could you replace the lost dopaminergic neurons with cells, with physical cells, right? So in the late 1980s, what they were using were small bits of fetal tissue from a 6 week to 9 week old aborted fetus. And they would cut out a piece of the tissue that would give rise to dopaminergic neurons, transplant them into the brain, and they would hopefully regrow the dopaminergic neurons. And it looked really exciting in the late 80s and early 90s. And then there were 2 large clinical trials in the US where nobody communicated. They didn't have the internet then, but they did have phones. Nobody communicated. Everyone used different doses of cells, different methods. Some people used immunosuppression. Some clinical trials used immunosuppression. Some didn't. Some of them just followed up the patients for 1 year. Some of them followed them a bit longer. And then some patients got something called graft-induced dyskinesias, and then everything stopped for about 15 years. But the patients that did well, did really well. So this is one patient from Sweden. This is a fluorodopa PET scan looking at the striatum. You should have 2 comma-shaped structures in health. So he got a unilateral graft of this tissue here, and this is time 0. In 1 year and 3 years after graft, his dopaminergic signal in his striatum was now better than healthy. It was back up to more than normal, and his symptoms improved, and he got off his L-Dopa. He was an effectively cured individual. So this is why there was so much excitement at the time, and there was so much racing, and a lot of egos got in the way, and everyone just messed it all up. Not everyone, a lot of people messed it all up. But 15-year hiatus, this is actually happening again. How many people know that fetal grafts are happening again in the UK? Does everyone know? Some of you know. Yeah. So that therapy that was happening in 1988, 89 is now happening again in the UK. And it's being led by Roger Barker in a clinical trial called TransEuro. The patient selection is very strict. Less than 65 years of age, less than 10 years of duration, minimal levodopa-induced dyskinesias. Sweden is also part of this trial in Lund, so they're trying to be really, really, really careful about all of the clinical parameters that were not cared for in the past, and they're looking at endpoint in 2020. But I can tell you about 16 transplants have happened already. About 8 patients have received transplants since May of last year, up until he gave an update at the World Congress in September. There may be more now. And these are, all from fetal tissue, right? So they're still relying on abortions. The abortions are happening in Cardiff or in Cambridge, and then they need about 3 fetal donations to treat one side. So to treat both sides, they need 6. And they do the surgeries, they're doing bilateral surgeries in Cambridge, and they do them at different times. They wouldn't do both sides of the brain at the same time. So they do 3. Several months later, they will then go in and then do the other side. But this patient did really well with a unilateral graft, right? So I don't know if he presented this in Portland, but he's had about 25 cancellations of surgery, and 15 of them were simply because there wasn't enough tissue. There weren't enough donations that happened in the 48. They have about a 48-hour window to collect those 3 embryos, and very often the patient's ready, everyone's ready, no embryos, so it's canceled. Okay, so what's the solution? So solution is, I and Roger and many people think that this can work for a number of Parkinson's patients, not all, but a number of them. But the solution, it can't be aborted fetal material. We can't rely on fetal material. So the solution, we think, is we need to make them in the lab. We can grow them, the exact tissue. We can make this tissue fairly reliably now in the lab without using embryos. And that will come from 2 very special types of stem cells called embryonic stem cells or induced pluripotent stem cells, which come from adults. And it really does avoid a lot of the ethical issues. It totally avoids the logistical issues. The logistical issues of having 3 fetuses in a 48-hour window is a nightmare. So we can define the product, and all the preclinical data in animals look really fantastic. They really do work. So I'm part of this Global Force Parkinson's Disease Consortium. It's maybe about 20 members, but it's trying to do the opposite of what happened in the 90s where no one communicated. Everyone who's going forward is communicating with this type of trial. And the three I think the three first trials that will happen, first probably in Kyoto. This is the Center for Induced Pluripotent Stem Cell Research and Application. So they'll use iPSCs, and that may happen next year. So I'm not talking 5 years down the line, this is next year. Lorenz Studer in New York will likely be in his first 5 patients in 2018. And then Roger Barker, Melon Palmer in the UK, Sweden is looking at shortly thereafter. So this type of therapy will be at least in phase 1 clinical trials in the not too distant future. So this is finally stem cells are going to do something, we hope, because there's been so much hype about stem cells for a long time, and this looks like it's going to be a really promising trial. I think that's it. Yeah, I'll stop there. And for those of you that go to the Congress, the next trip should be to Japan.
Is it true that the grafted cellular material acquires the disease itself unless there's some kind of suppressive treatment like using your alpha-synuclein vaccine in combination?
Yeah, that's correct, yeah. So I should have said also, in Kyoto is very timely, because I think by that 2019, some people may have received a graft in Kyoto. But will that graft get disease, is what Ken's asking. And the answer is yes. So that patient 4, who had that very successful graft, he died 20 years later, and his graft had Lewy bodies. So the graft is not immune to disease. It's human cells, it's human neurons, and Parkinson's is a spreading condition. But for most of the time he had this graft, he was off his medication. And it doesn't start getting disease for about 10 years. So some patients had died who had a graft for 9 years, and there was no sign of Parkinson's yet. So it does get signs of Parkinson's, but it's really slow. And Ken said the exact thing that we say now, but there's actually a way to make the cells disease resistant, which is what my lab is working on. So this is a cell therapy 2.0 what we're working on. It's very easy to make them disease resistant by one small genetic modification. But if these phase 1-2 drugs that we've been talking about do stop the spread of alpha-synuclein, these drugs, so you'd imagine you would get a graft, but then also get on one of these drugs to stop the graft from being invaded by alpha-synuclein. So the stem cell transplantation and these alpha-synuclein busting drugs could be used in combination.
My question is, in cell therapy, so what is the actual procedure? Do we first remove the affected neurons and then we transplant or graft fresh tissue, or just without removing the affected neuron?
Well, you don't need to remove them because they're going. In Parkinson's, the dopaminergic neurons are dying, right? So when you see a signal like this, there's much less dopaminergic neurons innervating that striatum right there.
So affected neuron, we are not taking out?
No. Well, you wouldn't want to take any neurons out. You need all of them. And this area, in some areas like the ultrasound-guided ablation, you probably are killing neurons. But you don't want to take these out. But you're asking because these neurons will spread the disease. Is that why you're asking?
Because means if affected neurons are already there, so if we are injecting new tissue, so what is actually happening there? Means
is there competition? Is what you're asking?
So because neurons are interconnected,
yeah.
So how they will make the new circuit and old circuit, how they will remove this?
Yeah. So they are interconnected. So there's sort of 2 types of neurons that we're thinking about. I don't have a picture of the brain, unfortunately, but there's a dopamine-releasing neurons that are made in a region of the brain called the midbrain. They have very long axons, and they innervate a place called the striatum. And that's what we're looking at here. We're looking at 2 striata, and this is where the dopamine is released. So they're innervating the striatum, but as the dopaminergic neurons are dying, they're not innervating the striatum, and then we try to look at the levels of dopamine, and the dopamine levels are going down and down in the striatum. But the striatal neurons, this is a different type of neuron, are still there. The striatal neurons are there, but it's the dopaminergic neurons that reach them that are gone. So when you put new neurons in there, they say, striatal neurons. They innervate them. They innervate them really well. Yeah. So the rat studies have been, I was going to put images of that from our lab, but if you put dopaminergic neurons in this striatum, they know to innervate them. And they will interact with the striatal neurons and start working. So if they're placed in the wrong area, and we have images of that as well, so sometimes in the rat experiments they're placed in the wrong area, such as the cortex, in a different part of the brain, they stay put, they don't send out axons. But when you put dopaminergic neurons in this region, they somehow know what to do, and they innervate. But they have to be immature. So what's transplanted aren't actually neurons, but they're called neural progenitors. So, which is why when they were using fetal tissue, this fetus had to be between 6 and 9 weeks old. If it was older, 11, 12, 14 weeks old, which is not a very old fetus, that tissue wouldn't work. It wouldn't innervate anymore, or they wouldn't survive the neurosurgical procedure. But they somehow know that they should innervate striatal neurons and start releasing dopamine. Yeah. 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 But this is really, really close to clinical trial. And the reason that we think this is going to work is there is already an existing fetal therapy. There's already a surgical procedure that's known. There's already clinical follow-up. So for 30 years, people have been transplanting cells in that region of the brain. And the only difference here is that we're not using fetal material, but they're lab-grown cells. Right? And this is so the final point is that, we've been talking about halting and slowing Parkinson's, but this is one that reverses Parkinson's. This is the only therapy that truly can reverse Parkinson's. Right? All the other ones, even the alpha-synuclein ones, the best that they can do is halt it in its tracks, right? And not have it progress anymore. The only way to reverse Parkinson's, at least the motor symptoms, is to rebuild the striatal network. And this will do that. This will rebuild the dopaminergic neurons.